Essay: Role of inorganic ions. An inorganic ion is a charged particle, that is, an atom or molecule which has gained or lost electrons. Ions have properties which provide functions in living organisms, which organic molecules are unable to do. For instance, ions are small and can be easily pumped across membranes, and they will readily diffuse down concentration and electronegativity gradients. This is a property which is used in several instances, which will be described later in later paragraphs. Inorganic ions are used to create potential differences across membranes because they are charged. In addition, by being charged, ions give important properties to biological molecules when they are part of the structure, as will be explained in the paragraph on hemoglobin and DNA structure. When ions bind to proteins, their charges interfere with the ionic bonding in the protein and therefore the tertiary structure, a property which will be explained in the paragraph on muscle contraction. A final function of ions is that by binding to molecules, they are effectively store energy, as will be seen in the paragraph on ATP. An example of ions being pumped across membranes and diffusing readily back down the concentration gradient is the uptake of glucose from the ileum to the blood. Sodium ions are actively transported from the epithelial cells of the ileum into the blood. This creates a sodium ion concentration gradient between the lumen of the ileum and the epithelial cell. Sodium ions rapidly diffuse from the ileum into the epithelial cells through co-transport carrier proteins by the mechanism of facilitated diffusion. Glucose molecules come through the protein at the same time, so they can be transported into the cell. Another example of ions being pumped across membranes and the diffusing rapidly down a concentration gradient is in the process of chemiosmosis, which occurs both in oxidative phosphorylation and in the light-dependent reaction of photosynthesis. High-energy electrons are transported along a series of electron carriers found in the cristae. Energy from the electrons is used to pump hydrogen ions across the membrane to form a proton-ion gradient across the membrane. Potential energy is stored in the gradient. The hydrogen ions diffuse back across the membrane through ATP synthase enzyme, which provides energy for ADP and inorganic phosphate ions to be bonded together to provide ATP. To transmit impulses, neurons have to build up a potential difference across their membranes. Inorganic ions are well suited to this function as they are small and charged. Neurons maintain a resting potential of 70 mV by actively transporting sodium ions out of the cell and potassium ions into the cell. Since some potassium ions leak back out again, there is an overall imbalance in positive ions, and the outside of the cell is positive in relation to the inside. This creates a potential difference across the membranes, and relies on the fact that the membrane is more permeable to potassium ions due to the leak channels, and less permeable to sodium because there are no leak sodium channels. Since sodium ions can diffuse rapidly across the membrane down a concentration gradient when the voltage-gated sodium ion channels open, then the electronegativity gradient action potentials occur. When the voltage-gated sodium channels open in the membrane, then there is rapid diffusion of the positively charged sodium ions into the cell, and this results in a change in the potential difference across the membrane as it becomes more negative on the outside than on the inside, making the membrane potential more positive. Within a few milliseconds, the potential difference is reversed again by the rapid diffusion of potassium positive ions out of the cell. This is because of the opening of the voltage-gated potassium channels and the voltage required being reached by the influx of sodium. As the potassium flows outwards, you get a brief switching in the potential difference across the membrane, and this is called an action potential. A wave of action potentials is propagated along the neuron axon, and this is called an impulse. Without small inorganic ions, organisms would be unable to transmit such impulses. Organic molecules can be given properties that they would be unable to have otherwise without the use of inorganic ions. An example of this is hemoglobin. Hemoglobin's role is to bind to molecules of oxygen in the lungs and transport them to respiring tissues. Only by having formed heme groups as part of its structure can the hemoglobin perform this function. Heme groups are iron ions. Iron ions very readily bind to oxygen molecules. Organic molecules would be unable to provide this property alone. Another example of inorganic ions providing properties to organic molecules is the presence of phosphate ions in DNA and RNA. DNA nucleotides consist of deoxyribose sugar, an organic base, and a phosphate. Nucleotides bind sugar to phosphate to give long polynucleotide strands. The sugar-phosphate backbone is very strong and stable and protects the sequence of bases. The presence of phosphate ions is essential to its function. The charge of phosphate ions is very important in the formation of membranes. Phospholipid molecules can only have a hydrophilic head due to the phosphate ion attached to the glycerol head of the molecule. The hydrophilic heads ensure that phospholipids form a bilayer which acts as a very effective barrier and controls movement of molecules in and out of the cells and allows the phospholipid bilayer to interact with water because of the hydrophilic nature of the heads. Inorganic ions, by being charged, interfere with the structure of proteins which are held in shape by ionic bonds. The binding of an ion causes disruption of the bonds and a change in the tertiary structure. An example of this is the calcium switch in muscle contraction. Sarcomeres contract when myosin protein heads bind to actin protein filaments. The heads then change shape and pull the actin filament to overlap with the myosin. The myosin heads can only bind when the binding sites on the actin are exposed. These sites are covered by tropomyosin protein. For a sarcomere to contract, calcium ions must be released from the sarcoplasmic reticulum in muscle fibers. Calcium ions bind to the tropomyosin protein, causing a change in structure, and the tropomyosin moves away from the binding site on the actin, allowing the myosin heads to bind and form a power stroke. A final use of ions is an energy store in the cell. The energy released from glucose during respiration is stored in molecules of ATP. ATP is an immediate source of energy to a cell for functions such as muscle contraction and active transport. ATP is formed by a condensation reaction between ADP molecule and inorganic phosphate. Energy is in effect stored in the bond and can be released to the cell when the bond is hydrolyzed. ATP is synthesized during glycolysis, the Krebs cycle, and as a result of oxidative phosphorylation. Only an inorganic ion can fulfill this role. All of the above examples of the use of inorganic ions emphasize that for processes to take place in living organisms, the properties that charged particles supply have to be utilized, as organic molecules cannot supply the same functions.